Now we are interested what's happening inside the heart. From myocardium, we move towards whole of the heart. So this is the heart. So let's make its four chambers like this are atria, these are ventricles. Okay, the whole of the atria acts as a syncytium. This means the electrical activity will goes around like whole of the atria. So from where the electrical activity starts, it starts basically from the SA node. So from the SA node the electrical currents are produced like this, and they will start moving, and as I have discussed earlier, the whole of the atria act as a syncytium. So these will move like this electrical. So we are not interested what's happening inside each and every cell, we are interested in what's happening in whole of the atria. Okay, so instead of each and every vector, if I draw a resultant vector, combined vector, it will be like small vector moving towards leftwards and downwards. So why it is leftwards and downwards? As SA node is present rightward and upward, so electrical activity will move downwards. So it will be like leftward and downward. So it is the resultant vector of SA node in the whole of the atria. So it is the electrical activity of atria actually. Now these electrical currents will not stop there. They want to depolarize whole of the heart. So there is something known as fibrous annulus. So fibrous annulus is like this blue one area. So it is not a good conductor. So it will not allow the electrical currents to move inside the ventricle through this path. The only path available for the electrical current is by AV node. So the current will enter into the AV node. Now let us talk some special points about this part, the AV node. So the main thing that is important about AV node, it is specialized tissue and it is very slow in conduction of electrical current. So let's consider this as AV node. So let's current has to pass from this point to this point. Okay. So if it has to pass, it has to pass to each and every cell, and it will show a lot of resistance. So due to this, there will be a slow conduction. So what will be the response of AV current passing AV node on the galvanometer? So because due to lot of resistance and slow conduction, there will be like electrical silence. So as we have seen earlier, like in resting membrane potential, there will be like this electrical silence. So in this case, like AV node, in the case of the ECG, we will see a electrical silence. Okay, in this case, we will not do any kind of vector. So let's consider this dark, consider, and there's no vector in that case because there's no detection of electric current by our devices. Now from AV node, the current has entered into the ventricle. So let's divide ventricle into three parts. The first one is interventricular septum. The second one is the apex of ventricle, and this one is like base of ventricle. Okay, so we will discuss each and every thing separately. So in the case of the interventricular septum, they are first discuss about two branches, right and left. So these are right and left. So basically both of them are insulated, and the left one is actually connected, and it is providing the electrical activity to the whole of the ventricular septum. So the electrical activity will starts moving like this, and will from this part to upward to the whole of the interventricular septum. The electrical activity will not be shown from the right side because right side is insulated and it is not actually connected with the interventricular septum. Only the left side is actually showing the electrical is giving the electrical current to the interventricular septum. So these vector, as we have discussed earlier, we are only interested in the combined vector of this whole of the interventricular septum. So let us draw a combined vector. So there will be combined vector will be like a vector, small vector moving rightwards and upwards like this one, because the depolarization starts from lower leftwards due to the left side of branch, because left side only giving depolarization, it will be like upward and rightward, and it is small because as compared to the whole of the ventricle, the interventricular septum is very small in area. But there is something special about this vector. Although this vector is very small, but as we have discussed earlier, their presence of Purkinje cells. Purkinje cells offer very low resistance, so this vector will be very fast. As we have discussed, if the this very fast, the galvanometer will show also deflection in very fast manner. Okay, as compared to the SA node, when it depolarizes the atrium, the vector was although big, but the conduction is slow as compared to the conduction present here at the interventricular septum. Now the depolarizing currents will move towards the ventricle. So let us discuss what's happened there. So there are right and left branches. So first we have to remove this apex and base world here. First discuss the apex. So from there, like this is endocardium, this part, and this is like subendocardial area. So first this area will be depolarized, then it will go like all along okay, so like this 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 and it, but as we have discussed earlier, we are only interested what's happening on each side. We will draw a combined vector for both right side and left side of the heart. Okay, let us make a combined vector. Let's remove this, make a combined vector for both. So let for the left side, the combined vector will be like this, and for the right side, the combined vector will be like this. As we know that the left side has to provide the blood to whole of the body, so left side has more thickness, left ventricle is more thick, so its vector will be bigger as compared to the right side, because its right side thickness is comparatively less as compared to the left side. So now we have to make a single vector for whole of the ventricle. So let's make a single vector for whole of the ventricle. So it will be like, so net vector will be like downwards and leftwards. It will be like this, because as we know both of the ventricles depolarization occur at the same time, so we combine both of the vectors. As we know, the left vector was far more stronger as compared to the right vector, because left ventricle has more thickness. So overall there the combined vector will be like this one. Okay, now we have discussed about the atria, the AV node, the interventricular septum, the ventricular base. Now the only thing left, ventricular apex, the only thing left is ventricular base. In the case of the base, the ventricles will be like, depolarization will be like upward and rightwards. So these are small vectors like upward and rightwards. So let us consider. So these are the all of the vectors of the heart, but there is something specific about the ventricle. All the ventricle vectors, although they are small or large, all of them are fast because there's presence of Purkinje cell in the ventricles. So the right, these are all the vectors present inside the heart. Now what we have done, we have connected a person with a galvanometer with negative, its negative rod connected with the right hand of the person, and left foot is connected with the positive side of the galvanometer, and we have connected the galvanometer with a paper, and we have made it to move. Okay, the paper is moving like this. Okay, paper is moving, and the pin of the galvanometer is on the paper. Okay, what we have done in short form, we have connected the person with the galvanometer, and we have connected the galvanometer uh, with a roll with a paper which is moving. Okay, now what actually is happening, we have a heart. I have copied that here. So in the heart we have seen that this, the first one was the electrical activity of atria, ventricle, and all of that. Okay, so basically the activity of the left side of the heart, left one and downward, was the activity of the ventricle. So actually what our body is a good, our body is a good conductor of electricity. As anything happening inside the heart, we can detect electrical activity throughout our body. So we have connected the electrodes with the arm and the left of the foot. So what's happening when there is no electrical activity, the we have also there, the paper is moving like this. When there is no electrical activity, the pin will be on 0 point of the galvanometer, so there will be a straight line. As paper is moving like this, there will be a straight line. Now what happening, Now when there is electrical activity in the atria, as we know the electrical activity will be in the atria, will be like this one, or if I draw there it will be like this one. So we have positive charge, positive electrode there, and electrical activity is also moving along that. So when, as we have studied earlier, when the positive vector is moving towards the positive electrode, so there will be a positive deflection. So as the atria is a slow conductor, so the velocity of the pin of the galvanometer will not be very fast, so there will be like this one. So it is electrical activity. Now this will be the electrical activity of atria. Now after passing through the atria, the current will move through the AV node. As I have discussed earlier, the electrical activity through the AV node is very slow and very small. So electrical activity, it is very small that it can't be detected by our ECG machine. So when electrical current passes through the AV node, so it will be like this straight line, because our ECG will not detect any kind of electrical activity when the current is passing through the AV node. Now after passing through the AV node, it will enter into the Purkinje cell, and first the active electrical activity will happen in the interventricular septum. So in the interventricular septum, the electrical activity is like, if I draw the positive and negative, the electrical vector is this vector is fast, small, but is moving away from the positive. So this is positive and it is moving away from the positive. So if it is moving away from the positive, electrical activity deflection will be against. So first deflection, like if the needle is moving upward, now it will start moving downward. So because this is also very fast, so needle will move fast. So deflection will be like this one, small but very fast vector. Okay, so this is interventricular depolarization. Now after this depolarization of the ventricle, so the vector for the depolarization of vector was big vector, a strong vector moving downward and leftward. We have also attached the electrode on this side, so it is moving towards the positive electrode, so deflection moving towards the positive. And as I have discussed earlier, all the vectors that are formed in the ventricle are, the deflection shown the galvanometer is very fast because there is presence of Purkinje cells. Okay, now if we draw it, there is strong fast, so it will be a strong, a big vector, and very fast. So it is representing the major depolarization of the ventricles. So in the last, the thing is that is that has left is the base of the heart. So if we saw this vector, these are very small vectors, and these vectors are moving away from the positive, so they will show negative deflection, as in the case of interventricular septum, but these vectors are smaller. Okay, so if we draw them, this will be like this. So it is ventricle, so this will be fast deflection. So if we see this, this was P, this was Q, this was R, and this was S. So let us revise. So first there was atrial depolarization, the vector was moving downward, leftwards towards the positive, so it was a positive deflection. But because atria was not a very fast conductor, so there is a formation of P wave. After this, the when the current passes through the AV node, there is no deflection. After this, the current passes through the interventricular septum, so the vector is moving away, but the vector is very fast and small, so there is negative deflection in the form of Q wave. After this, there is major ventricular depolarization, the vector is towards the positive, and it was a big vector, so is R wave is formed and it is very fast. So after this, the last are the bases, so these vector are moving away, so there is negative deflection, and this is the ventricle, so there is very fast deflection, so the S wave is formed. Up till now we have discussed about depolarization only. Now we will discuss about repolarization. So as we have discussed earlier, the cell which was last to be depolarized is the cell which was first to be depolarized. So the last cell which was depolarized was from the subpericardial myocardium. So like depolarization will start from here. because it was the last to be depolarized. So it will goes on like this and goes like this way. So if we draw a vector for this, so the vector, the major ventricular depolarization will be like this one. It will be like this one. It will be like upward and rightwards, like this one, upward and rightwards, and at the same time. So but the main thing about this vector is this vector is not positive. As we do all of the vector, it's actually the negative charge that is moving upward and rightward. So this is like negative, not positive. Instead, a negative vector that is moving upward and rightward. So so let us draw the repolarization vector of the ventricles. So for the ventricle, the repolarization vector will be like this one. But the main thing is all the vector we do were positive vector going towards the positive. This is actually a negative vector, because when there's repulsation occur in the cell, the negative, it will goes towards the resting membrane potential. In that case, the negative, the anions are more inside the cell as compared to the outside. So it's the negative charge. So repulsation is actually negative vector. So negative vector is moving towards the negative charge. In other cases, in case of deposition, positive vector were moving towards the positive, so show they were showing positive deflection. In this case, their negative vector is moving towards the negative charge. It will also show us the positive deflection in ECG. So let us draw. So one main thing is that repolarization is very slow process, although it is happening inside the ventricles. And as we have discussed, in the case of repolarization, the repolarization process in the ventricle was very fast, but repolarization in the ventricle is very slow. So if we draw the repolarization, so in ECG it will be like this one and this. So this is T wave, while between the S and T, the whole of the ventricle was depolarized. This was the before the T wave, that was the part when repolarization wasn't started yet. So let us summarize all of the ECG that we have studied till now. So first was the atrial depolarization. The vector was positive vector moving towards the positive electrode. So and it was slow. So it was showing a positive deflection in form of P wave, and the vector was slow. So it will form like a dump. Okay, after this, the electrical activity passes through the AV node. So AV node was showing uh, electrical silence. After this, there was depolarization by the interventricular septum, and the vector was uh, not along the positive electrode, it was moving away from it. So it was also very fast vector, so it was showing Q. After this, there was major ventricular depolarization, and it was shown in the form of vector, a big vector moving very fastly towards positive electrode. So R wave is formed. After this, the depolarization of the base occur, and it is also moving against the positive electrode, so it is showing negative deflection, and it is very fast, so it is showing S wave. After this, the whole of the ventricle is depolarized, so it was galvanometer was showing a straight wave, like no deflection. After this, the repolarization occur, and repolarization is very slow process, and its vector is negative vector moving towards the negative electrode, so it is also showing a positive deflection in the form of T wave.